Today we're going to talk about the Magnoliophyta, the flowering plants. And it's important that we spend a whole lab talking about them because they represent over 90% of all the embryophytes, and that's over 250,000 species of known plants. So clearly they're quite successful. They've evolved and diversified into a much broader variety of habitats, and in fact taken over and pushed out other plants like the pine trees that used to occur in a more larger geographic range on the planet. So we're going to focus on the Magnoliophytes, the flowering plants, and not only do they have flowers, but they have fruits. And they have another special adaptation called double fertilization. We'll talk in a little more detail about that, but all three of these adaptations enhance reproductive success. So flowers are going to attract pollinators, and the pollen is going to get moved from one flower to another at a higher percentage than it would without the pollinators. So having flowers is going to be an adaptation that increases fertilization success. The probability of any particular egg getting fertilized is going to be higher. The purpose of fruits is to help disperse the seeds. So remember last time we talked about the fact that the fruit is the ovary of the plant, the seeds are inside of it. And what needs to happen in order for this species to persist and thrive across the landscape is that the seeds need to get a little further away from the parent than just falling right on their feet, because then they would be in direct competition with the parent. Now, there's good habitat where the parent is, but it's possible that there's good habitat elsewhere that this species would like to colonize. And also the habitat might change, and then if the parent plant dies, then if the seeds dispersed a good distance away, then the species can persist because it isn't in that bad habitat anymore. So because the plant packages the seeds inside of something that's interesting to animals, now they can be dispersed in other ways, water and wind as well, but many plants make a fruit that an animal's going to eat. The seeds aren't destroyed by passing through the animal's digestive tract, and so they get moved because the animal can do locomotion away from the parent plant. So this is called dispersal. So fruits are going to enhance dispersal, right? So the movement of those seeds away from the parent plant. And that is going to, in the long run, increase success and survival of the species. The third adaptation, double fertilization, actually has two sperms. So instead of one sperm fertilizing an egg and that's it, you're done, there's a second sperm that fuses together with some other cells inside the female gametophyte, and it produces a triploid tissue called endosperm. Endosperm is what nourishes the developing embryo inside the seed. So there's two reasons why that's important. One is it gives the seeds a good start in life. Basically, the seed is filled with nutrients and energy molecules, just like your mom packs you off to school with a lunch, hopefully. And so the other thing is that production of that seed is not triggered unless fertilization happens. So it saves the resources of the mother plant. If the seeds aren't fertilized, the eggs aren't fertilized, then she doesn't produce the fruits. All right, so we'll look at that in more detail again later. So the grouping of plants within the Magnoliophytes is fairly complicated. This slide is just up here for me to remember to tell you that when we talk about plants and we talk about them being dicots versus monocots, this isn't all of the flowering plants. So we have monocots that are going to be in this grouping, and then these eudicots are everything else. But there's some interesting other plants that are off on this little twig. We're not going to talk about them, you will in a botany class. And they include interesting species like Magnolia, which is an actual flowering plant that grows its seeds in a cone. So it's kind of an in-between between truly flowering plant with an ovary covering the seeds versus a cone plant. It's a cone plant with a flower. So when we look across all these different plants, we can divide them into different families. For example, we have the pea family. There's hundreds of species, if not thousands of species. And so we have the rose family. So plants with many similar characteristics we group together. And we're going to talk a little bit more in this lecture about how we tell apart monocots from dicots. When we're talking about flowering plants, we want to look at the structure of the flower. The structure of the flower has a ring of petals around it, and there are also sepals that are part of this ring. You may not be able to tell the difference between the sepals and the petals, which make up the corolla or the crown of the flower. The sepals are just defined as the ones that are farthest on the outside and cover the bud when it's first formed. They may or may not be green or brightly colored like the petals are. So you have to see the plant flower developing to be able to tell which is which. Many flowers have both male and female reproductive parts inside of them. So the female part is going to be this ovary that's all the way in the middle. It's going to have the ovules inside of it. And it's going to have a stalk that sticks up, the tip of which is called the stigma. Now the stigma is sticky, and the point of that is because that's where pollen grains are going to land. And it needs to be a place where the pollen grains can settle down and then germinate and start to grow down to where the ovules are. So the carpel overall is this female reproductive structure. It has three parts: the stigma, which is the tip of the carpel, the style, which is the stalk, and the ovary, which is a usually hollow shaped structure at the base. Right? Sometimes it's above the sepals and sometimes it's below them. The male reproductive structures inside the flower include the filament, which is just a stalk, and then the anther is at the tip. The anther is going to produce the pollen, and together these parts are called the stamen. You can remember that the stamen is the male part because it has the word men in it. So an important feature of these flowering plants is that they have cooperation from animals in order to perform pollination. So pollination is the transfer of pollen from one flower to another, often on a different plant. So that allows genetic diversity to be produced within a population. And the different flowers have evolved over time to match together with the characteristics of their pollinators. So particular pollinators have preferences for different kinds of characteristics of flowers that they visit. The actual shape and size of the face or the structure on the pollinator has to brush up against the place where the pollen is on the flower. So there's incredible diversity of different types of pollinators. You might think of bees, you might think of hummingbirds, but bats are also a very important pollinator, a variety of kinds of, all kinds of different flies, wasps. There are mammals can be pollinators, marsupials are pollinators in Australia. And different ways the flower can attract those pollinators include the scent, the color. Many insects can see different color range of light than we can. They can see UV, so sometimes there are patterns that show up on the plant. Sometimes the flower pretends to be something that might attract the pollinator. Some flowers look like female insects, and the male comes and he thinks he's getting a hot date, and he's actually pollinating a flower. So they often give a reward, however, to the pollinator. So nectar or a sugary liquid can be released by the flower in order to attract a pollinator as well. The time that the flowers are open during the year, the time of day the flowers are open, is going to also be related to their pollinator. So bats are flying around at night, so those flowers may be open at night and not be open during the daytime. When we look at different flower characteristics, like animal bodies, we can characterize flowers by the kind of symmetry that they have. We can also characterize flowers by whether they include both male and female reproductive structures or only one. So the vocabulary for these characteristics of flower structure are that if the flower has radial symmetry, that is, we're looking at the front of the flower, and we see that the petals are basically the same all the way around. So the flower has radial symmetry. We're going to say it's radial symmetry, it's regular, right? A regular flower. Now, if it's irregular, that means the petals aren't all the same shape, and usually this flower has bilateral symmetry. So we're going to be able to draw a line down the front. All right, so an example of a irregular flower is an orchid. An example of a regular flower is a daisy. When we look at flowers and we see that the inside they have both carpels and stamens, that is, they have both male and female reproductive structures, we say they're perfect. All right, so stamens and pistils in the same flower. We can also have plants that have different flowers on the same plant that are separate sexes. So they may make male flowers first and then the female flowers after. Some species we can have where each individual plant is only a male or a female. In that case, you need to have more than one tree of whatever kind it is in order to get fruit on your female tree, because you need to have a male around. So either the flower is perfect, it has both stamens and pistils, or it is pistillate, that is, it has the female parts only, or it's staminate, which has the male parts only. Right, so equivalent vocabulary when we're talking about animals is monoecious versus dioecious. So if the plant is monoecious, it has both stamens and pistils. It's like being a hermaphrodite. Typically, they're going to have adaptations to prevent them from fertilizing themselves, however. Other differences there can be in flower structure have to do with how the petals are arranged and how many different flower parts there are. So simple flowers are basically going to be kind of like our flower diagram. They're going to have those reproductive structures in the middle, and then they're going to have a ring of petals around the outside. We can have these simple flowers, which is basically saying there's only one flower in this flower. All right, if that doesn't make sense, you'll see what I mean. There's something different that's happening in the sunflower. So we have a simple flower, and it can either have petals that are fused together. So if we look along this location on this morning glory, we see that the petals can't be separated from one another. Over evolutionary time, they fuse together. So the flower is a continuous tube, but we can see that there used to be 1, 2, 3, 4, 5 separate petals on this plant. Now, if we look at a different plant, California poppy, we see that each petal has a separate outline, and you can pull them off one by one, so those are separate. Now, if we don't have a simple flower, the opposite of a simple flower is a compound flower. And a compound flower, or sorry, a composite flower is actually compound. That is, it's a flower made of many flowers. So we have these, what looks like petals around the outside edge, but those are actually also a special kind of flower. And when I say flower, that means they have their own little pistils and stamens inside them. In the middle, there are hundreds of tiny single flowers. They're called disc flowers. So when we look at these composite flowers, they're actually hundreds of separate little flowers, each with their own stamens and pistils inside of them. If they all get pollinated, this sunflower, each one of those little tiny flowers is going to make one seed. So this head of all these sunflowers we see is because they have all of those different little tiny disc flowers separately, each one making a seed. Right? The aster family is the most successful large family of flowers. It's part of the dicot, so sunflowers. If you see a daisy-looking flower, then you need to know that it's a composite flower, that it's a dicot. Sometimes you can't tell just by looking at the flower parts what group it's in. So once the flower has died, the eggs have been fertilized, the ovary is developing into a fruit, the ovules are developing into seed, we're going to see there's also incredible diversity in what kinds of fruits are built by this process. So one of the easiest things to do is tell the difference between what are called dry fruits, that is, they're woody and they aren't something like this seed pod that you're going to want to bite into. So another example of a dry fruit is something like a walnut or a peanut that has that dry, hard husk on the outside. And a fleshy fruit is one that you're going to be able to bite into and is often juicy and delicious. When we look at the way that the fruit develops from the parts of the ovary, right? So this is something that you usually can't tell just by looking at it. If you're in a botany class, you'd have to memorize which fruits were made of which different parts. We're not going to worry about that too much. We can have different structures that end up becoming parts of the fruit, and that botanically they're built somewhat differently from one another. But we're only going to worry about dry fruits versus fleshy fruits. And often these fleshy fruits are going to be ones that are going to be attractive to animals, whereas these dry fruits are going to get transported by wind or by water. So the way that the ovaries form into the fruit can also be different between different plants, because we can have a fruit that's made out of a single ovary that has turned into a single fruit, or we can have multiple flowers, multiple ovaries that have each formed a seed, and then the whole thing becomes fused together. So if we have multiple ovaries inside one flower, or if we have more than one flower that ends up contributing to making that single fruit, we have a compound fruit. So again, we have simple fruits versus compound fruits. Simple fruits are majority of different fruits. So for example, you think about that avocado with that one big pit in the middle. It was made from one ovary from one flower. However, if you look at strawberries or pineapples, all these kinds of little parts that they're made of are actually parts of different flowers or of different ovaries that fuse together, right? So this is another thing that you need to know how that fruit develops to see it from the time that it was a flower in order to know. But I just would like you to know a couple of examples. So these are pretty easy. If you remember that most of these berries and pineapple are compound fruits, that's all you need to know. So when we're looking at these fruits, we see that they can be transported by animals, but that's not the only way, by eating them, and that's not the only way that they can be transported. Sometimes they'll actually stick onto the outsides of animals that have some little Velcro-y like hooking structures, and as the animal passes by, they'll stick onto them. They can get blown by the wind if we have milkweed or dandelion. This is the fruit, and it's got this big fluffy structure on it. It gets picked up by the wind and blown. Some seed pods are capable of sort of violently exploding and spraying their seeds all over the place. Some seed pods are at the top of long stalks, and when the wind blows, they get snapped off and thrown quite far. We have been domesticated by plants in order to propagate them. You can think of that as one way of thinking about agriculture, that we have some types of food plants that take over an incredible amount of land area, and we carefully take care of them. So we actually propagate those seeds, disperse those seeds for those plants. Mind control those. And plants that live near water, typically their fruits float, and that allows them to disperse rather than just drown. All right, we're going to dig just a little bit deeper into the flowering plant life cycle. This is a review of alternation of generations, and we looked at seed and pollen last lecture. That we have the megaspore is going to be found inside the ovule. The ovule is going to be the female gametophyte is going to be 8 cells. It's going to be inside the ovule. The egg is going to be one of those cells. The pollen is going to manage to get to that egg somehow. And then we're going to have fertilization happen inside the ovule. And then that zygote is going to develop into an embryo. So the ovule is going to become the seed, and the ovary is going to become the fruit. There's a little more detailed description of what happens here. So here we are looking at the base of flower, and we have these chambers of the ovary down here. So these little dots are representing the ovules. So meiosis is going to happen there. We're going to get these megaspores. We're going to get some mitosis happening and some cells dying, and we end up with this 8-celled female gametophyte. This is inside the ovary still. So one of those cells is the egg. All right, it's right down here. And it's going to be at the very bottom of this structure. Now, somewhere else, there's a stamen that's producing microspores that are going through, meiosis producing microspores, and then they're going to release those pollen grains, and the pollen itself is the male gametophyte. It's only 2 cells. So a pollen grain is going to land on top of this stalk, and it's going to land on the sticky stigma. And what it's going to do is basically break open, and one of the cells is going to grow a tube that's going to penetrate down through the tissue of the style, and it's going to grow down and around, and to the point where the sperm are going to be able to go down this tube. So we're going to have 2 sperm. The other cell that starts out inside the pollen is going to divide into 2 sperm. So those 2 sperm are going to go down to the very bottom of this embryo sac. So the embryo sac is another term for this gametophyte. So notice there's 8 cells here. There's the egg, there's these 2 cells on either side of it. Then there's 3 cells at the other end, and then there's these 2 cells in the middle, this big empty looking space here. All right, so we're going to have 1 sperm that's going to fertilize the egg. I notice that little yellow dot there. And then we're going to have the second sperm is going to bypass the egg, and it's going to go into this middle region of the embryo sac. And so there's three haploid nuclei here. And so we're going to end up with the 3N situation of this tissue that ends up in the middle of the embryo sac. And it's going to grow into a tissue called endosperm. Endosperm is going to be like when we look inside of a chicken egg. We have all that yolk, we have the white, that's the place for all the nutrients that are going to support the developing embryo. That endosperm is the energy and nutrients that are going to support the developing plant embryo. So the double fertilization doesn't happen in pines or other non-flowering and non-seed plants. It is unique to Magnoliophytes. All right, that is the review of reproduction in the Magnoliophytes. We're also going to look very quickly at differences between monocots and dicots in the structure a little more detail, partly because we can look at flowers in order to tell us the difference. So just a quick reminder that we have a difference between monocots and dicots, and that they get their names because they get one little leaf that comes up out of the soil, or if they're a dicot, when they germinate, they have two little leaves that are going to come up out of the soil. We're going to look at their tissues for the stem and the roots. So we're going to look at some cross sections at stem and at root, and we're actually going to see that the pattern of how the vascular tissue looks is different between monocots and dicots. So just looking at a cross section of the stem and then seeing the pattern of the vascular tissue, we can tell the difference between monocots and dicots. We are also going to look at a little bit about what their leaves look like as well, and then their flower parts as part of the lab activity. All right. So when they're growing, the way they grow down into the ground, so dicots tend to have a large and long taproot that's very thick, whereas monocots may have a lot more thin and fibrous roots. When we look at the stem cross section, when we look at a dicot, we're going to see this ring of vascular bundles. And when we did our slides earlier in the semester of our staining activity, we were looking at a dicot stem. And so we did see this pattern on those cross sections. Now, when we look at the stem of a monocot, we're going to see that those vascular bundles are scattered or at random. All right, so we'll be able to look and tell the difference because of the pattern that we see in vascular bundles. All right. In order to talk about the way that these patterns work, sometimes it's useful to have one more vocabulary word, and that is the brown tissue that's in the middle region here is called the pith, right? And the region that's outside the vascular bundles, outside, she said, trying to draw on the slide. This region that's outside the vascular bundles is called the cortex. All right, so we see that there's a very, very thin cortex in the monocots, and that the pith is also much smaller. If we look at the root cross section, right, in the root, the vascular tissue is centered near the middle, the very middle of the stem. And when we look at how it is patterned in the dicot, it's going to be an X marks the spot in the very center. So not only can we tell the difference between dicots and monocots, we can tell the difference between root and stem, right? And I'm going to expect you to be able to do that. Now with a monocot root, we do not have an X. We have this pattern where we have a circle in the middle, and then to me it looks like a sunflower with a little array of petals around it. We have, in both cases, we have extensive cortex, but there is no pith, right? There would have to be a place inside towards the center of the vascular tissue, and the vascular tissue is in the center, so there is no pith in the root cross sections. All right, so we can tell monocot from dicot root, we can tell monocot from dicot stem, and we can tell the difference between stem and root. All right, when we look at the patterns of leaves, we're going to be able to see where they have their veins. So just like in our circulatory system, when we have vascular tissue in the leaves, we are going to call those veins, and we can see them on the surface of the leaf, right? So we're going to look for the veins on the surface of the leaves for monocots versus dicots. And we're also going to look at a cross section of the leaves. So in monocots, we're going to tend to have those long, thin leaves. We're going to have parallel veins. And so if we take cross section across the leaf and we look at it on the end, we'll see the veins are all the same as one another, and they are just in this row here. So this is a cross section of those parallel veins. Now, when we look at a dicot leaf, we see that there's this branching. So there's some kind of V's or intersections between the veins. We don't see this parallel pattern. So this one on the right-hand side is dicot. And they tend to have this large, thick mid-vein. Sorry, accidentally closed the slideshow. Let's just get back to it. So they're going to have this large mid-vein, typically. And when we look at a cross section across the leaf, we'll see that there will be this sort of V shape. And then when we're seeing these smaller lateral veins, they may look a little strange in cross section because we're seeing them at an angle, right? So they're passing crossways, sort of a little angle here, right? But we'll still be able to see clearly that there is vascular tissue along the leaf. And it's not in this nice, neat, tidy pattern where everything is the same size. We're going to have that one much larger vein. All right, so there is lab activities that have you look at these differences between monocot and dicot. There's lab activities to also look at the patterns of the flowers of monocots versus dicots. So monocots are going to have, for example, their petals. Usually you'll have 3 or 6, whereas the dicots are typically going to have 4 or 5, right? But the leaves are the very easiest way to tell the plant. You can use the leaf vein pattern even if the plant doesn't have any flowers on it right now. And then we're going to have a bunch of slides where you're going to get to see the series of steps that happen inside that embryo sac and how it starts to develop into a seed.